Hello guys, so today we're going to analyze blood group in practicals. We have questions here that I took from our quiz from the genetic module. So if we look at question 1, we have Forrest, patient B there, has 3 children with his partner Lila. Crystal, this is Crystal here. Then we have Crystal is 19 years old, Raven, and Scarlett is patient E. Crystal has been learning about blood groups at university and decided she wanted to know her blood group. Her father paid for the family to have their blood group at a private lab, as he secretly suspected that their family friend Moss, patient A, so this is Moss, and this is Forrest. I'm just gonna call Forrest father 1, and this guy is suspect F2. Okay. So Moss to be the father of their youngest child, Scarlett. They included blood samples of the youngest child and from Moss. Lila is the mother, already knew her blood group A+, from when she was tested during pregnancy. Antibodies specific from different antigens on the red blood cell surface were added to each blood sample. Following incubation, researchers looked for evidence of agglutination in each reaction. The results are here, so we can see. So to explain what these numbers, what these positives and negative mean, I'm gonna have to go down here to explain it to you. Okay. So this is what happens when you want to find out the group, the blood type. So what we do is, there's a slide. We have antigens, antigen A and antigen B. One end of the slide is labeled antigen A and antigen B. So there are serums here that contain antigens, What will happen is a blood sample is dropped on both antigen A and antigen B. Then, depending on what that group blood type is, what will happen, one of the antigens is either antigen A or B will have agglutination. So agglutination, this is what it looks like. It's when you have blood crumbles or they cluster together and the color would change sometimes. So for this specific slide, or this blood that we tested on both of these, we've used a blood sample from the same patient, but we tested it with antigen A and antigen B. So in this case, we can see agglutination happening within antigen A. So that tells us already, it's very straightforward and easy. If agglutination takes place with antigen A, then its blood group type is A. So it will be A. But if it happens with B, then it's B. And if it happens with both, then it's AB. Okay? So to look at examples, real examples, here we are. So question 1, we have antigen A and antigen B, and we have used a blood sample on both of these. You can already see in antigen A, the blood still looks the same. It's still red, no no results, nothing is expressed, and agglutination has taken place with antigen B. And so moving on to question 2, we can see that on antigen B there's no difference. We can still see the blood and it looks exactly the same, but there is agglutination with antigen A, which tells us this patient blood type is A. If we go to number 3, we can notice a difference in both of them actually. So when there is difference in both of them, that tells us straight away it's AB. And of course in number 4, as you know, the group O is a recessive one, so there is no agglutination at all in both of them. Yes, you can see maybe this is a bit pinkish, this is a bit darker. Yes, it's okay. But the main important part is when you see a big massive difference, such as here in A or in B, how you can see like a very apparent difference. So this group type is this patient blood type is O. So now we've learned that if there is agglutination in antigen A's, so that means it's patient with group blood A. If there is a difference, or if there is agglutination expressed in antigen B, it's B. This one is A. If there is agglutination in both, it's AB. And if there's no agglutination, of course it will be O. So now you have it there, the four blood group types and how they look actually on a slide. So now going back to our question, now we have to find out or figure out the blood group types for each of our patients, patient A, B, C, D, and E. As we can see here, the test has been done. For antigen A, we have positive, but what does that tell us, a positive? Here it says in the question, positive denotes agglutination present and minus means no agglutination. So when we have agglutination in A and B, that means it's an AB. We have no agglutination in A, agglutination in B, so that's group B. We have agglutination in A, so that's blood group type A. In B, in none of them, because these are related to those, so that's group O. Now you might be wondering, what about this one? Okay, so to go to this one, now we have to learn about antigens T. Okay, so the RH system, RH antigens, they are also surface antigens that are expressed in red blood cells. There are few RH antigens. Common one is called D. So antigen D, you will see that a lot in different questions. So red cells are RH negatives. Okay, so where is it? Red cells expressing RH antigens are called RH positive. When we say expressing, that means agglutination. So when we have agglutination, that means RH is positive. When we don't have it, that means RH is negative. So going back to the question here. We are going to now add the RH group for each. So for the first one, we said his blood group was AB and his antigen D is negative, so that means RH is negative, so minus basically. For patient B, we have positive, so agglutination, that means it's RH positive. And this goes the same for these. And now we have basically figured out the different type groups depending on the results we have been given. Now, if we continue to question 2, it asks us, what is most blood group type genotype? So we are now looking deeper into blood group types, and we're going more further into genotypes. So if we go back to the original question, the first question, Moss is actually patient A. So his blood group type was AB minus. So he's AB minus. So whenever we have to address blood group types, it's very good to have this in mind over here. Okay. So this table right here, it's something that we need to memorize, especially this part. This part is very important in genetics. It only tells you what are the genotypes that we, like for example, genotypes for O, as I said before, O blood type is actually a recessive type, so it's not always expressed. So the genotype for it is always I. It's very simple and easy to remember. It's just the small, it's not capital case letters. For type A, there's 2 different types. We have this one and this one. We have I capital A, or I capital A, small case I. And the difference is this one is from a dominant parent. So when both parents have A blood group types. This one is one of the parents will have an O. So what you see, it's when you have a small I, that means IO as in a recessive one, same as in type O. For type O blood, you have small case I's. So for this guy, we said, for example, if you have type A and it's the recessive one, like here, that means one of your parents passed an A to you from their blood type and the other has passed an O. But because A is dominant and O is a recessive, the traits will show as A, and it will act as A when it's tested because it will be causing agglutination in antigen A. So as I said, these groups you're meant to actually, like the genotypes, you're meant to memorize. They are not very hard, they are fairly easy. It's very just important to remember if you have type A group blood, blood group, so you have two genotypes, two possible genotypes. If you have group B, you have two possible genotypes as well. If you are type AB, you have only one outcome, the same as type O. You only have one outcome. So in type AB, it would have been one parent given a dominant A and one is passing a dominant B allele. So when these alleles are passed, they become the combination to form a blood group type AB. So again, these are not related to the recessive part where, the RH. These are not related to it. So if this is this one here, this is a recessive one, but it still is called blood type B. So it's very easy just to remember. It's okay, it's capital B for B, or a capital B and nothing. Capital A for A, capital A or nothing for A again. Just small letter I for O, and A, capital B for AB. So these will not change depending on the RH factor. The RH factor, the factor is inherited independently from the ABO blood type. Genotypes for the RH factor are this, and this. Those three. So if you have this one, then your RH is positive. Also, if you have this one, RH is positive. If you have this one, your RH is negative. So these are genotypes. The way it's written here, it shows as a minus. So 1 minus from a father, 1 minus from a mother. A plus from a father, a plus from a mother, a plus from a father, and a plus from a mother. So these are genotypes. Remember, these are genotypes. So it's not actually, when we ask what is the RH type, you don't write this way. You only write it this way. That way. Those three ways. So remember why, for example, if you're wondering, okay, so this one in the middle, it has both. It has plus and a minus. So how come it's actually called RH positive? Well, it's because the positive RH is actually dominant. So although we have a negative factor, RH factor, but remember that positive RH factor is more dominant. So if we go back to our questions. Okay, so we said his blood group type is AB, and remember we said this has got nothing to influence the genotype of letters that are written like that. So we said AB, regardless, it can't be recessive, so the answer is this one. For question 3, from the information provided, is Moss Scarlett's father? So Moss, we said he's an AB minus. Supposedly, this is father, and mother is A plus. Okay, and if we go to Scarlett is O positive. So we're saying, can he be the father to this child? So we have those two blood group combination, and we're asking ourselves, can it give us this one? So if we come here, we have this table here. So it tells us the possible combinations. It's almost like blood type calculator. So we said Moss was, actually let's go to the father. Moss was an AB, regardless whether positive or negative, and the mother type was A. So that gives us those possible outcomes, and none of them show an O. So how could you memorize this table when you are in exam, because you don't have access to it? So what can you do to find the answer quickly? So it's fairly simple to just realize that we're gonna outline this right now. Okay, so if we look through this line, including this line, there is no O blood type being produced. Why do you think that? That's because whenever you have an AB with an AB with an A, you can see an AB with an A gives you AB or AB. There's no O. An AB with a B, there's no O. AB with an AB, there's no O. AB with an O, even an O itself, you still do not produce an O. That's just the first rule. So an AB will never ever produce an O. That's what you can notice. And you can see almost in all the AB interactions, most of them, they produce those three outcomes: A, B, or AB. A alone, B alone, or AB. Except when it's actually interacting with blood group O. So that is the first rule, or the first thing to remember, is that AB will never ever produce an O. Second thing, an AB with an O will only produce two outcomes, an A or a B. Then we can move on to interactions with A blood type groups. As you can see, there are so many types. So within A, I guess these things you have to really remember. When an A is bonded to a B, it produces 4 types. When it's bonded to an AB, it will produce all 3 types. When it's bonded to an A or an O, it produces the same exact result. So when it's bonded to itself or bonded to an O, it produces an A or an O. When it's bonded to a B, you have 4 outcomes, almost all the groups. When it's bonded to an AB, have A alone, B alone, or an AB.